So we're currently in the biochemistry, officially in the biochemistry portion of the course. We're going to see that the chapters in these modules are different than the chapters from organic chemistry. In organic chemistry, each unit of study dealt with a couple of different functional groups and more analysis of them. Here in biochemistry, it's very different because biochemistry is essentially organic chemistry writ very large. We have many functional groups, perhaps in a type of biochemistry we're looking at. It's all organic structure, but it is truly the chemistry of life in that it's in a much larger sort of disposition in which the molecules we're going to be looking at are more complex, are larger, but will ultimately be based upon those organic functional groups that we saw before. Notably, so what we're starting with now is going to be carbohydrates. Carbohydrates are a class, if you will, in biochemistry, a class of types of molecules, and we'll talk a little bit more about what that means. But no longer are we talking about a specific functional group. Rather, we're talking about a structural type. Carbohydrates have a particular umbrella under which certain structural types we see, and really, if we thought about it, the most simplest way we can describe carbohydrates we'll be looking at, or I'll be terming them as, is sugars. If you'll notice on some of these molecules, here's a representative carbohydrate. We're going to see what that is. We have a number of functional groups, many of them on that particular molecule. For instance, on this molecule, we have a number of, for instance, alcohol or hydroxyl functional groups, many of them on one molecule. We didn't see that in organic chemistry. We kind of kept it nerfed up, kind of simple, but we have, in this case, 5 hydroxyls, 5 alcohol functional groups. We have an aldehyde, which is a carbonyl, flanked on one side by hydrocarbon-carbon, the other side by a lowly hydrogen. So we have this many different functional groups coexisting on the same covalent molecule. So that's where we're going to be looking at with these different units of study under the auspices of biochemistry. There are 3 levels of structure to carbohydrates, which will encompass this unit of study. The first one is the simplest type, that is considered the monosaccharide, also colloquially known as simple sugars. Monosaccharides are the smallest monomer building block of what could be considered higher levels of intricacy. In nature, we see the basis of a lot of biochemistry as being based on the polymer. We've talked about polymers before. Each polymer has to have a monomer unit that is repeated throughout. As we go through that, the smallest monomer in nature's polymers of carbohydrates are the monosaccharide, and we spend most of our time talking about that. The second level of intricacy to carbohydrates would be considered the disaccharide, in which we have 2 monosaccharides that come together to form what are considered dimers. Dimers of monosaccharide units, di for 2, mers for polymer. So it's really a polymer of 2. These are colloquially known as complex sugars, but essentially they're the covalent attachment of 2 monosaccharide units, and that'll be the second area we study in carbohydrates. Lastly, we're going to be talking about what are considered polysaccharides. Polysaccharides, colloquially known as complex carbohydrates. These are true polymers, true nature's polymers that we're going to see over and over in different units of study within biochemistry, but they're essentially polymers of what are those monomers? Well, the original ones, the monosaccharide units that we will begin studying right now. Monosaccharides are the most basic form of carbohydrates. As we said before, they are considered simple sugars, and they have the general atomic ratio of 1 part carbon, 2 parts hydrogen, 1 part oxygen. Indeed, CH2O. So where the term carbohydrate comes from, we have carbo for carbon and H2O, water, for hydrate, carbohydrates. Down below, we see some representative renditions of carbohydrates, specifically the monosaccharides, given in a very specific way. We see these carbons, these carbon atoms in all of them is given a sort of a zipper effect down the center with all of the oxygens and hydrogens set off to the sides. There's a specific method of doing this to represent the monosaccharide in its linear form. If we look at there's two ways we can look at monosaccharides. The first would be in what's called a Fischer projection, and we're going to look at that first. A Fischer projection is written always in a particular way. We have our carbons down the center. We have our oxygens and hydrogens off to the sides. It's a specific way that is the way that shows the most information one can get from a molecule of a monosaccharide. We find that larger monosaccharides, and by larger I mean between 5 carbon monosaccharides or 6 carbon monosaccharides, these considered longer linear sequences, can exist as a cyclic form as well. So we have our linear form as well as a cyclic form, and with a cyclic form is always given in a particular way as well. It's known as a Haworth projection. The Fischer projection of a monosaccharide, as I said, gives more information, structural information, and it's something we're going to be looking at first. Fischer projections are a way of expressing molecules in all of organic chemistry. Specifically, we're going to be looking at Fischer projections with monosaccharides. This Fischer projection is always shown as a linear chain of carbons. As we see in all of these cases, we have a linear chain of carbons right down the center in any Fischer projection of a monosaccharide. That's part of the two criteria that Fischer projections ultimately have. If we look at these two criteria for a Fischer projection, and we're just looking at it with respect to monosaccharides, the first is that all of the carbon atoms in our Fischer projection of a monosaccharide are arranged in a vertical line down the center, as I said, like a zipper. And if we look at on any monosaccharide, a linear projection of a monosaccharide, there's always going to be what is considered a more oxidized carbon, more oxidized. So if we look at all of our carbons in here, most of them will be carbons connected to have one carbon-oxygen bond attached to them, so connected to the hydroxyl in these alcohol units as well. But there is one that is considered most oxidized, and that would be the carbonyl group, which is evident on every single monosaccharide in its linear form. That's C double bond O. In a Fischer projection, the most oxidized carbon is always located or situated towards the top of the Fischer projection. Not always the top carbon, but towards the top. So here now we see in all of these molecules, in this Fischer projection of this monosaccharide, it is that most oxidized carbon shown by that carbonyl is the highest, is the top carbon. On two of these, we see that it is definitely the top carbon. On this center structure, we see the most oxidized carbon as not the top carbon, rather, but it is, if we were to situate this one way up, one way down, we would have to have that carbonyl situated towards the top. So given in this direction here, we see this line of carbons would have to be set such that this carbonyl is set towards the top. So in all of these now, we get a particular way of expressing them, such that there's two rules here. We have the first being that all carbon atoms are arranged in a vertical line, and with the most oxidized carbon towards the top. While we're not looking specifically at nomenclature, or rather at organic chemistry, we have a way of classifying monosaccharides. Now, classification in organic chemistry was when we looked at primary, secondary, or tertiary sort of structure. Now that we're in biochemistry, this concept of classification means something a little different. With regards to classifying monosaccharides, we're doing this not necessarily to name them, a nomenclature, although we will see that in the future, but rather a way of sort of putting them into different baskets, so of arranging them. So linear monosaccharides, and they have to be expressed as Fischer projections, can be classified into groups by two types of structural features. Now, the first would be whether we have our carbonyl carbon always on any monosaccharide. Here's a carbonyl carbon here, carbonyl carbon there on this Fischer projection of a monosaccharide, always situated towards the top. It depends on what kind of carbonyl we're talking about. In this case, we're looking at the structure on the left. That is an aldehyde functional group. Therefore, this molecule this monosaccharide is expressed or classified as what is considered an aldose, for aldehyde. We'll find that this suffix OSE, ose, is the suffix for any carbohydrate, be it a monosaccharide, a disaccharide, or a polysaccharide, will always have the suffix OSE. So this is classified because it has an aldehyde as its most oxidized carbon here as an aldose. Notice that's not a name. We're not talking about nomenclature, or rather we're just classifying it. The structure over here on the right-hand side, its carbonyl, is a ketone. It is a ketone carbonyl based upon the type of functional group we're looking at. Therefore, it is considered a ketose because it has a ketone as its most oxidized carbon. So we have aldose or ketose, which is one way in which we can express or classify a monosaccharides without naming it. The other way to express them, to put them into baskets, to classify monosaccharides in the Fischer projection are as follows. So it depends upon how many carbons are contained within it. If we look at the structure over here on the left, we have how many carbons? 1, 2, 3. Thus, it is considered a triose as a classification. Tri for 3, ose as because it's a carbohydrate. Next one over here, if we have 4 carbons, 1, 2, 3, 4, it is considered a tetrose. We have a little contraction there. Tetra is typically the Greek prefix for 4 carbons, but we contract it rather than tetraose, it is a tetrose. Moving along, a 5-carbon monosaccharide in the Fischer projection, pentose, a 6-carbon monosaccharide in the Fischer projection, as we see here over on the right-hand side, hexose. Again, we're not giving names to these, we're just classifying these. So, a classic exam or a quiz question might be to show these sorts of things. Which one is the aldose? Which one is the pentose? And so on and so forth. Now, we're going to find that in this classification pattern that we can put these two structural features together, whether the aldehyde is or whether the carbonyl is an aldehyde or ketone, as well as how many carbons are contained within it, into one type of classification. And this would be combining these two to give more precise terminology. For instance, if we look at this molecule over on the left-hand side, we see that it has an aldehyde as its functional group, as its most oxidized carbon, and we have 1, 2, 3 carbons in it. So it would be considered an aldotriose. Takes that aldehyde as well as the fact that there's 3 carbons within it. It's an aldotriose. Here is an example of a monosaccharide in a Fischer projection form that has 1, 2, 3, 4 carbons, and that ketone carbonyl makes it a ketotetrose. 2 terms within that classification. The next one we see here, well, that has, let's look at that most oxidized carbon. That is an aldehyde, and it has 5 carbons. It would be considered an aldo for aldehyde, pentose for 5 carbons. And lastly, over here, it's sort of obscured by my picture here, we see that is a ketone functional group over on the right with 6 carbons within it. It would be a ketohexose. So we could look at this. I mean, once we get these two very simple ways of expressing it together, we can move forward. And if we're presented with some problems like we see here, we could very easily look at these and classify them with both of those structural features. The first, if A in the upper left-hand corner, would be given as a ketotetrose, because as a ketone, most oxidized carbon and 4 carbons within, ketotetrose. For B, we find that we have an aldehyde as well as 1, 2, 3, 4, 5 carbons would be an aldopentose. C, 6 carbons with a ketone, ketohexose, and we just see moving forward with these. Once we understand the basis of this, we can very easily classify these using both of these types of criteria. Since biochemistry is at a much higher level of complexity than organic chemistry, we now have to encounter or speak to a type of isomer that was not necessarily apparent when we were looking at organic molecules, but does become apparent with a higher, as we said, level of complexity in biochemistry. To review, let's look at our types of isomers that we talked about in organic chemistry. There was our constitutional isomers, which had the same molecular formula and different atom connectivity. There's a couple of examples right here. We then moved into the geometric isomers, as a review, had the same molecular formula, same carbon connectivity, but different spatial orientations of substituents. When we were talking about geometric isomers, we looked at situations where we had a pair of compounds. Perhaps it would always have a cis and trans type of moniker put onto it. But now we're going to start looking at a new type of isomer brought on by this complexity in biochemistry, that of what is considered stereoisomers. There's no real better way to express or show one's the stereoisomer concept than to look at what we all have as this particular model kit, that of your own hands. If we look at our two hands, I have a right hand and a left hand. They both have 4 fingers and a thumb, and if we look carefully at it, if I go up to a mirror and I put my right hand up waving at it, it's going to be my left hand waving back at me. Now, we know that our right hands are not our left hands, because even though we have 4 fingers and a thumb on each hands, they're clearly different, which is why we have a right hand and a left hand. We can find that our right hands and our left hands differ in that they are considered non-superimposable mirror images. If I took one hand and tried to place the other hand over it, I cannot superimpose my both hands together. I cannot superimpose them. My thumbs are sticking out to the side, and but what I can do is superimpose over each other as what is considered a mirror image. So our right and our left hands are considered stereoisomers in that they have the same molecular formula, meaning 4 fingers and a thumb, the same carbon framework, the connectivity. They're both put together similarly. The spatial arrangements are identical. If we have we have 4 fingers and 2 thumbs set like that. However, they are different, and they are set apart because they are considered non-superimposable mirror images. My right hand is the mirror image of my left hand, but I cannot superimpose them. My thumbs sort of stick out to the side. Now, it's very fanciful to express the concept of stereoisomers to a pair of hands. It's a very apt way of doing it. However, we really want to look at stereoisomers and how they apply to actual organic molecules. Can you have 2 organic molecules that would be considered non-superimposable mirror images? And it turns out that you can, and we'll see that on the next slide. In this slide, we see a pair of organic molecules. In this case, they are considered stereoisomers because they are non-superimposable mirror images. Now, if we'll notice, we have a carbon atom on each of these. If we look at that carbon atom, right in both of these cases, we see they each have the same substituents emanating from them. But they're in different colors here to sort of express the ability to reflect, if you will, from a mirror plane, which we see right in the center here. You'll see that these molecules are written three-dimensionally. Tetrahedral carbon is three-dimensional, so that the purple balls right here would be both edged back. Those are both expressed as mirror images. The red balls on top here, the A ones, are also similarly reflected. And the yellow and the green balls on this model are similarly reflected. Now, it's a little harder to see in this when we're talking about organic molecules, but in fact, these are two non-superimposable mirror images. In fact, if you laid both of these models across one another, we would not be able to exactly superimpose one over the other. They are considered stereoisomers. Now, when we look at organic molecules specifically with respect to stereoisomers, I know we used the example of my hands or your hands in the last slide. When we refer to specifically organic molecules, stereoisomers between them are considered to be known as enantiomers. Enantiomers is just another way of saying stereoisomers. However, at it specifies organic molecules. This is one of the molecules from the enantiomeric pair on the previous slide, and I want to make a little commentary on the carbon atom, the central carbon atoms within there. They are considered, they have a special consideration to them in that they are considered chiral carbons. A chiral carbon. How do we tell what a chiral carbon is? Well, it has two criteria, really. The first one is that all four bonds emanating from that central carbon are single bonds. And indeed, we see that. These are four single bonds right here. So that criterion is taken care of. Perhaps the more important criterion is that all four of those single bonds must terminate in different structural environments. Now, that can mean the same atom. It can mean the same carbon group, that sort of thing. In this case, they're all given by the different colors of these terminating atoms on the end of it. So that this carbon atom in the center would be considered to be a chiral carbon. Now, molecules often have more than one chiral carbon, and it becomes important to be able to express or to identify carbons in a molecule, asking whether each carbon by carbon, is considered chiral or non-chiral. If we look over at the molecule on the left-hand side, there is one central carbon in this case, and we can say that it is indeed a chiral carbon because all four bonds emanating from it are indeed single bonds. And if we look at what they terminate in, we have a hydrogen, a chlorine, an oxygen, and a bromine. So those are four different environments. So that central carbon is considered a chiral carbon. If we look at the next molecule over, we have two carbons in this one. The one up top, let's look at this carbon up top. I can immediately say that that's a non-chiral carbon because it has two of the same substituent, two hydrogens attached to it. All four substituents on those single bonds have to be different environments. Two hydrogens are the same environment, so this is indeed a non-chiral carbon. If we look at the other carbon in this molecule, in this case, it is going to be a chiral carbon. Why might that be? Well, here we have hydrogen, chlorine, oxygen, and indeed another carbon attached to it. Four different environments, a chiral carbon. Looking at the third molecule in the series, we can really just pick these out based upon what we've done thus far. So if we look on the top carbon on here, this carbon right there, well, we have, in this case, just like the last time, we have hydrogen and hydrogen. So those are two of the same environment, so this would be a non-chiral carbon. If we look at the bottom carbon, let's go with the bottom carbon now, we see it is four separate covalent bonds connected to hydrogen, chlorine, bromine, and carbon. Thus, that is indeed a chiral carbon. So we have a chiral carbon here now. Lastly, let's look at this, this last carbon right there. I am considering that to be a chiral carbon. And why might that be? Well, let's look at what we have connected to it. We have a hydrogen on the left, an OH on the right. Here we have a carbon on top and a carbon on the bottom. Let me clear up this piece so that we're actually looking at this specifically. This central carbon right here is connected to hydrogen, oxygen, carbon, and carbon. Now, why wouldn't that be non-chiral if we have the carbon connecting to two identical carbons? It turns out those two identical carbons attached to that central carbon are not identical. This one, let's look at one in this case, is this carbon right here is a CHCl-Br carbon. This carbon up top here is this carbon connected to hydrogen, bromine. So these two different carbons on the top and on the bottom are in two different environments. So thus, this would be a chiral carbon in the center here. It's a little hard to see because they're both carbon atoms that are connected to, but we have to take that connectivity further to look at for any differences that we can find. Now, most importantly, and the one type of structure we're going to be looking most of all at whether there's chiral carbons in it are specifically monosaccharides. And here is a monosaccharide in the Fischer projection, and we can immediately go through these and express whether we have chiral carbons or non-chiral carbons. If we look at the most oxidized carbon in here, the one on the top, remember, on the Fischer projection, we see this carbon right here is immediately considered non-chiral, and the reason for that is we have a multiple bond on there, C double bond O, immediately. That's non-chiral because remember, all four bonds from a carbon have to be single bonds. Let's go to the bottom one, the bottom carbon. Immediately I see two identical environments attached to that carbon, so this is also non-chiral. Now, if you look at the two remaining carbons, they are both considered chiral carbons, and let's look at why that might be. Let's look at this, the carbon on top right here, this carbon right there. Actually, what we'll do is we will move that to a different color. This carbon right here, a better one. Let's do a better one. We'll do green. This carbon right here is connected to hydrogen, oxygen, CHOH carbon, and C double bond O carbon, different environments, thus chiral carbon. Similarly, on the next carbon down that we see right here, we have hydrogen, oxygen, CHOH carbon, CH2OH carbon. This carbon is also chiral. It takes a while to figure this out on these things, but it is indeed doable. A classic test or quiz or an exam question might be show the chiral carbons on this compound, or that sort of thing, and we'd be able to sort of pick through that and be able to identify all those carbons as chiral or non-chiral. We're going to find that our bodies recognize frequently, not all the time, but frequently, different enantiomers of the same molecule. Two different molecules of a stereoisomer pair we can perceive differently. For instance, Carvone is an organic molecule which has, as we see, two superimposable mirror images, two enantiomers, or rather two stereoisomers. One stereoisomer, our olfactory and taste buds represent that it's a smell of and taste of spearmint. But the other enantiomer, it tastes like caraway, like the seeds in rye bread. This is the result of it associating differently with the olfactory receptors in our nose or our taste buds in our mouth. A different one, and more ominous really, is an old drug that used to be used in Europe in the 50s for birth control. One enantiomer treats morning sickness very well, but it turns out the other enantiomer actually caused very grievous birth defects in the women that were using the drug to treat the morning sickness. So in this case, the body recognizes the difference in chirality, or the difference in action of enantiomeric pairs. Now, it is important at this point to go into a little more detail on what the meaning is when we talk about Fischer projections. Remember, Fischer projections were set on a grid sort of thing. We had horizontal lines, horizontal bonds and vertical bonds, and what that actually means. Because we're dealing with molecules which have 4 single bonds emanating from a common carbon, that's tetrahedral carbon, and that is 3-dimensional. So we have to be able to show 3-dimensionality. If we look at the enantiomeric pair set over on the left-hand side, which we show right here, we see that these models are built in a 3-dimensional fashion. Now, this is written sort of in a quasi-Fischer projection. At least the bonds are either straight up and straight down, or side to side. When we do that, which is inherent in a Fischer projection, always the bonds which are vertical, the vertical bonds in these cases, always 3-dimensional wise, tip back into the plane of any overhead or any picture we're looking at. Any horizontal bonds tip out of the plane towards us. That is always the case. So we can see that in both of these structures on the left. If we were to draw a line structure, say, of this molecule pair right here, that's just in 2 dimensions. But since it's in a Fischer projection, we can associate these horizontal lines with wedges. They're tipped out of, this is what it actually looks like in 3D, or expressed as in 3D, tipped out of the plane of the page. And these vertical lines always tipped back into the plane of the page. That is always a specific consequence of Fischer projections. Now, granted, these Fischer projections are not on monosaccharides, but in the next slide we will look at how that actually expresses itself in monosaccharides. On this slide we have 2 different versions of pairs of stereoisomers on an actual monosaccharide. Notice on the pair on the left we have a standard Fischer projection with its attendant vertical and horizontal lines. What that really means from the chiral carbon in the center of this of these pair of monosaccharides on the left is shown on the pair on the right. Here we have our chiral carbon, and it actually shows the depth, the 3-dimensionality of this pair of monosaccharides. This is going to become important when we designate monosaccharides as 2 different types. Only one particular carbon, a chiral carbon, is going to be of importance in this case, and we'll take a look at that in order to designate 2 different ways of looking at monosaccharides. Monosaccharides can be designated as 2 different types, D or L, when we look at them in a Fischer projection. It's very important that a monosaccharide be in the Fischer projection in order to carry out this designation. There is a process for doing this, and we're going to find out what D and L ultimately mean, and how to look at any monosaccharide in the Fischer projection and be able to designate it as one of these 2 things. The first thing is that we have to find the chiral carbon. Notice I have that underlined in capital letters, because as we know, there can be more than one chiral carbon on a monosaccharide. In this case, the chiral carbon on a monosaccharide is the chiral carbon located furthest from the most oxidized carbon on a Fischer projection. So if we look at the molecule, the Fischer projection of a monosaccharide on the left-hand side, here is the most oxidized carbon. That's the one with the carbonyl on it. And we have to find the chiral carbon which is furthest from it. It's always going to be below it. It is not the bottom one, because that one, CH2, is not chiral. It would actually be this one. In fact, in this molecule, there's only one chiral carbon, and it is the furthest one, it's the only one, from that most oxidized version. So this is considered the chiral carbon. Once we locate the chiral carbon, we look at which direction the hydroxyl, the OH group, is pointed on it. In this case, the hydroxyl is pointed to the left. Therefore, the L in left means it is an L monosaccharide. This monosaccharide is of the L designation. If we look at the molecule on the right, this monosaccharide, in this case, we locate the most oxidized carbon. That's this one, always, the one of the carbonyl. And we're looking for finding the chiral carbon. That's the chiral carbon located furthest from the most oxidized one. Well, we have 3 carbons below that carbonyl. The bottom one, again, is not chiral because it has 2 hydrogens attached to it. We have, as a possibility, this one is chiral and this one is chiral. It is the one that's furthest away from the most oxidized carbon that is considered the chiral carbon. In this case, the hydroxyl points to the right. If it points to the right, it is a D monosaccharide, a D monosaccharide. So we can literally look at any monosaccharide in the Fischer projection and designate them as D or L. Now, importantly, in nature, only D monosaccharides are synthesized, are used, are metabolized, never an L monosaccharide. Nature is very one-sided in that manner. Sort of a classic question is shown here. Indicate whether the monosaccharides shown are either of the D or the L designation. So we're looking for D or L on these. And really, in each of these cases, we have to find the disposition of that hydroxyl on the chiral carbon. In this case, the chiral carbon furthest from the most oxidized one. On A, we'd have this one. B would be this one, C this one, and so on and so forth as we move along. And on these chiral carbons, we look, basically just ask, which direction is that hydroxyl pointing? In this case, in the case of letter A, it is pointing to the right. All of the ones that are pointing to the right here, I will highlight in yellow, and indeed, these would all be of the D designation. There are only 2 left where the hydroxyl on the chiral carbon is pointed to the right. That would be B and F. And in this case, both of these would be considered L monosaccharides. So as you can see, we have a situation where, knowing these 2 very simple rules, any monosaccharide given to you in the Fischer projection, its designation as D or L can be ascertained. We are going to stop this lecture here, and we'll see you in the next lecture.